Title: Adaptive Integration Enables Evolutionary Rescue from Extreme Environmental Pollution. Authors: Elias Oziolor, Noah Reed, Sivan Yair, Kristen Lee, Sarah Guberman-Verplag, Peter Bruins, Joseph Shaw, Andrew Whitehead, and Cole Mattson. Abstract: Radical environmental change that provokes population decline can impose constraints on the sources of genetic variation that may enable evolutionary rescue. Adaptive toxicant resistance has rapidly evolved in Gulf killifish, Fundulus grandis, that occupy polluted habitats. We show that resistance scales with pollution level and negatively correlates with inducibility of aryl hydrocarbon receptor AHR signaling. Loci with the strongest signatures of recent selection harbor genes regulating AHR signaling. Two of these loci introgressed recently, 18 to 34 generations ago, from Atlantic killifish, Fundulus heteroclitus. One introgressed locus contains a deletion in AHR that confers a large adaptive advantage, selection coefficient S equals 0.8. Given the limited migration of killifish, recent adaptive integration was likely mediated by human-assisted transport. We suggest that interspecies connectivity may be an important source of adaptive variation during extreme environmental change. Main text. Human alterations in the environment can be swift and severe, and thereby result in population declines and increased selective pressures. Adaptation requires genetic variation from mutations, standing variation, migration, or interspecific hybridization, integration. However, declining population size coupled with immediate threats to fitness may constrain the sources of genetic variation that enable evolutionary rescue. New mutations arise slowly. Standing variation may be insufficient, habitat fragmentation may limit migration, and hybridization may be rare and deleterious. Little is known of the relative importance of these sources of variation in the context of evolutionary rescue. Gulf killifish, Fundulus grandis, are common in coastal estuaries along the northern Gulf of Mexico. They occupy the Houston Ship Channel, much of which is heavily polluted with halogenated and polycyclic aromatic hydrocarbons, HAHs and PAHs respectively, resulting from more than 60 years of industrial activity, forming a gradient of contamination through Galveston Bay, Figure 1A. Many HAHs cause cardiac deformities in developing vertebrates that directly impair fitness. Nevertheless, Fundulus grandis persist at these sites and are resistant to the normally lethal effects of these toxicants. We examine the molecular underpinnings of apparently evolved resistance to extreme pollution among populations densely sampled across the pollution gradient, using experimental and population genomic approaches. We integrate prior work on the sister taxon of Fundulus grandis, the Atlantic killifish, Fundulus heteroclitus, which has also adapted to similar chemicals, to uncover the evolutionary history of key adaptive variants. We sampled fish from 12 sites spanning the pollution gradient, Figure 1A and Table S1, spawned them separately in the laboratory, and exposed their embryos to a range of concentrations of polychlorinated biphenyl 126, PCB 126, a model HAH. At the population level, resistance to toxicity, as measured by rates of cardiac teratogenesis, is correlated with pollution along this gradient, where fish from the most polluted sites are resistant to concentrations of HAHs 1,000 times higher than normally teratogenic levels, Figure 1B and C. Resistance is retained through at least 2 generations in a clean environment, Figure S1, and is intermediate in hybrids from crosses between sensitive and resistant populations, indicating a genetic basis. Aryl hydrocarbon receptor, AHR, signaling mediates HAH-induced cardiac teratogenesis. We compared AHR pathway function, CYP1A, activity in response to PCB126 exposure, Figure S2, among populations across the pollution gradient. Similar to resistance, desensitization of the AHR pathway scales with levels of pollution, Figure 1D and Figure S3. Within intermediate populations, inter-individual variation in AHR inducibility ranges between sensitive and resistant populations, Figure 1E. Our experimental results suggest that desensitization of AHR to HAHs underlies resistance in Fundulus grandis. To understand the genetic basis of this phenotype, we sequenced whole genomes of 24 to 49 fish, approximately 0.6x coverage, from 3 resistant, R1, R2, and R3, 2 intermediate, IH1 and IH2, and 2 sensitive, S1 and S2, populations. We searched for signatures of selection that co-vary with pollution through pairwise genetic differentiation, FST, population branch statistic, and differences in nucleotide diversity, pi, between polluted and sensitive populations in 20 kilobase windows. Neighboring 20 kilobase windows were merged into regions which were ranked by their size and overall divergence. We defined the 1% most differentiated windows as outliers for each polluted population. Genome-wide differentiation between populations is low. Pairwise FST equals 0.002 to 0.029, figure S4, and genetic structure is consistent with geography, figure 2A. Compared with sensitive populations, resistant populations have reduced nucleotide diversity and increased Tajima's D, figure 2B and figure S5, possibly reflecting a recent reduction in population size. Evolved resistance, figure 1, accompanied by strong recent selection, figure 2C and figure S6, and population decline, figure 2B, is consistent with evolutionary rescue in polluted populations. Top-ranked signatures of selection are shared across all pollution-adapted populations, figure 2C. We detected 88 shared outlier regions distributed across 23 of 24 chromosomes. Differentiation in shared regions is higher between resistant and sensitive populations than between intermediate and sensitive populations, figure 2D, and intermediate populations show no consistent signatures of selection relative to both resistant and sensitive populations, figure S7. We conclude that intermediate populations are distinguished from resistant populations not by selection on different loci, but rather by weaker selection at the same loci. Genomic regions showing the strongest signatures of selection harbor genes encoding key regulators of the AHR signaling pathway. AHR1A and AHR2A are centered in the most highly ranked shared outlier region, chromosome 1, figure 2C and figure S8. Two paralogs of aryl hydrocarbon receptor nuclear translocator, ARNT, which dimerize with AHR to activate transcription, figure S2, are within the second and third ranked shared outlier regions on chromosomes 8 and 10, respectively, figure 2C and figures S9 to S11. Aryl hydrocarbon receptor interacting protein, AIP, regulates nuclear translocation of pollutant-activated AHR and is within the R1 to R2 shared outlier region on chromosome 2, figure 2C and figure S12. Signatures of selection in multiple AHR pathway elements, coupled with additional selection signatures throughout the genome, indicate that AHR pathway modification is an important component of polygenic adaptation to the fitness challenges present in this urban estuary. The top-ranked outlier region spans approximately 2 megabases and encompasses the tandem paralogs AHR1A and AHR2A, figure 2E. We detected a 77 kilobase deletion spanning these genes at high frequency in resistant populations, 85%, 83%, and 68% in R1, R2, and R3 respectively. At moderate frequency in intermediate populations, 25% and 5% in IH1 and IH2 respectively. And in only one heterozygous individual from a sensitive population, figure 2E and figures S13 and S14. Given that the AHR pathway is profoundly desensitized in resistant populations, figure 1, D and E, and figure S3, and moderately desensitized in intermediate populations, and that its experimental knockdown is protective of toxicity, we propose that this mutation is important for the adaptive phenotype. 5 different deletions in AHR genes have now been associated with rapidly evolved resistance to HAHs across 3 species of wild fish. Consistent with the less is more hypothesis, this suggests that rapid adaptation by disabling mutations may be common. We speculate that sweeps of such large effect loci may quickly recover fitness after extreme environmental change, whereas smaller effect loci, but with reduced negative pleiotropy, advance more slowly. We analyzed whole genome sequences from Fundulus grandis and Fundulus heterocolitis to contrast variants associated with resistance. In resistant Fundulus grandis, the region containing the AHR deletion on chromosome 1 as well as the region from chromosome 10 encompassing ARNT, was more similar to Fundulus heteroclitus haplotypes than to other Fundulus grandis haplotypes, figure 3a and b and figure S10, which could result from incomplete lineage sorting or admixture. The species divergent time estimated from elsewhere in the genome predated the divergence of the selected haplotypes, figure 3c, suggesting admixture as the source. To formally test for integration and to estimate the timing and strength of selection, we extended a coalescent theory-based inference method that distinguishes among modes of adaptation. Integration of the deletion-bearing haplotype was much more likely than incomplete lineage sorting, figure 3e and figures S15 to S17, consistent with divergence time estimates, figure 3c. We inferred a large fitness advantage for individuals carrying the AHR deletion haplotype, S equals 0.8, and very recent gene flow, 16 generations before onset of selection, figure 3e, with the sweep occurring over 18 generations. This model estimates that integration happened approximately 34 generations before sampling. As we are unlikely to have sampled the true Fundulus heteroclitus source population, resistance is widespread in this species. This time may be even shorter. For the adaptive ARNT region, we infer similar timing of gene flow, 36 to 37 generations before sampling, and strong selective advantage, S equals 0.55, figures S18 and S19. These alleles clearly have a large effect on fitness and so likely explain a large proportion of the genetic variance in pollution tolerance. We conclude that recent integration from a few successful Fundulus heteroclitus migrants, figure S17D and S19D, provided crucial genetic variation to rescue Fundulus grandis populations from rapid environmental change. We evaluated the footprint of integration in Fundulus grandis genomes by scanning for genomic segments greater than 400 kilobases, showing evidence of Fundulus heteroclitus ancestry. These ancestry blocks collapse into 15 discrete regions totaling 70 megabases on 9 chromosomes, figure 3a and figures S20 and S21. Most large integress blocks are rare. 11 of the 15 regions are found in fewer than 5 individuals, and are found exclusively in polluted populations, figure 3d. More than 30 megabases of the 40 megabases comprising chromosome 1 showed evidence of integration in at least one individual, representing Fundulus heteroclitus ancestry that hitchhiked with the deletion sweep. Mean haplotype block size also suggests that the integration event was recent, approximately 65 to 155 generations before sampling. A recent discrete hybridization event occurred in the Houston ship channel, introducing highly advantageous adaptive haplotypes from Fundulus heteroclitus and leaving behind evidence of Fundulus heteroclitus ancestry scattered across the genome. Because Fundulus heteroclitus have small home ranges and the nearest populations are in Florida greater than 2,500 kilometers away, human-mediated transport is the likely mechanism of introduction, possibly through ballast water transfer or bait fish transport. The importance of hybridization in conservation biology is contentious. Recipient populations are at risk for negative ecological interactions with invaders, introduction of locally maladaptive or deleterious alleles, or genetic incompatibilities. We propose that Fundulus heteroclitus integration into Galveston Bay was sufficiently rare to preclude extensive accumulation of deleterious variation in Fundulus grandis, and that the adaptive advantage afforded by integress loci was sufficient to overcome maladaptation imposed by linked loci. Although a growing body of work shows that ancient hybridization can contribute adaptive genetic variation, our work shows that hybridization can provide variation crucial for adaptation following swift and extreme environmental change. I'm now going to walk you through the 3 figures illustrated in the main text. Figure 1, variation in sensitivity to pollution among Fundulus grandis populations distributed along a steep pollution gradient in Galveston Bay, USA. Panel A, pollution gradient is scaled by color from low, blue, to high, highly polluted, black. Populations include resistant, black, R1 to R3, intermediate high resistance, red, IH1 to IH3, Intermediate low resistance, gold, IL1 to IL3. And sensitive, blue, S1 to S3. Genomics data were collected for populations denoted with circles. So you can see here we've got multiple populations distributed along this pollution gradient from clean, Galveston Bay, Outer Bay, to really polluted within the Houston Ship Channel. This is a fairly short geographic distance across which these populations are distributed. And it is from the circle populations that we collected genomics data, but we have phenotype data from all of these populations. Panel B, population variation in cardiac deformities in embryos exposed to PCB 126. Error bars indicate standard error of the mean. You can see here that populations differ in their sensitivity to increasing log doses of PCB 126, and here we're scoring the deformities that are caused during embryogenesis upon exposure to these different doses. And as we can see, populations sampled from the blue sites are sensitive at much lower doses than populations sampled from the more polluted sites. Population variation in sensitivity to PCB-induced cardiac deformities, log mean effective concentration EC50, correlates with, in panel C here, habitat pollution, and in panel D, AHR pathway inducibility, CYP1A activation by PCB126. So again, panel C here, we've got pollution along this scale here, measured for each site, measuring the sediments from each site. And here is the effective concentration that causes cardiac deformities in the animals from each site. You can see that there's a fairly strong correlation here. So populations' sensitivity or resistance seems to be scaled by the level of pollution. As we can see in panel D here, we've got a negative correlation between your sensitivity and your CYP1A activity. Basically, more sensitive populations have greater latitude of inducibility of CYP1A than the resistant populations. Panel E, PCB-induced CYP1A activity varies among individuals and populations. So we can see here basically the variation among populations that we see in panel D, but also expanded to see the variation among individuals within those populations. So similarly, here we've got the very resistant sites. They've got the lowest CYP1A activity. Sensitive sites are CYP1A activity is much more inducible by exposure, whereas our intermediate populations sort of span the range of inducibility between the sensitive and the resistant populations. Figure 2, genetic variation within and among populations. Panel A, neighbor joining tree from genome-wide data indicates population clustering along the pollution gradient. Okay, so here we used all of the population genomics data, so genetic variation from across the genome, to unite individuals and populations that are most genetically similar. And as you can see, populations that are sampled from geographically nearby locations tend to be genetically most similar. And that's consistent with sort of our neutral expectation that genetic similarity should scale by geographic distance. Panel B, consistent with a demographic bottleneck, populations from polluted sites have an excess of rare variants, which is to genus D, top panel, and reduced nucleotide diversity genome-wide, pi, in the bottom panel here, compared with clean site populations. Elevated to genus D is consistent with recent population bottleneck, as is reduced pi that we can see in the resistant site populations compared to the sensitive site populations. Panel C, genetic differentiation for each resistant and intermediate population, for these 5 panels here, compared with both sensitive populations through the population branch statistic. Background divergence is low, and purple indicates outlier regions, 1% most divergent, that are shared among all adapted populations. So here we are scanning across the genome. Here we're comparing genetic differentiation between the R1 population compared to the two sensitive populations, both of them. Okay. And here we see genome-wide, there's not much differentiation, but there are these big peaks of differentiation here that are signatures of selection, recent strong selection. Here we see for another resistant, the third resistant population, and for the intermediate resistant populations compared to, again, all of these are compared to the two sensitive site populations, two sensitive populations. And what we can see here are these divergence regions that tend to be shared for each population compared to the sensitive populations. Panel D, multidimensional scaling MDS of genetic variation for shared outlier regions clusters individuals and populations by resistance level. Okay, so here we're grabbing these loci that are the shared outliers, these purple ones, and we're asking, how do those regions of the genome distinguish genetic similarity or differentiation between individuals within our analysis? We can see here that intermediate populations are distinguished from the sensitive populations, but the resistance populations are quite differentiated, even more differentiated from the sensitive site populations. Panel E, a deletion in Fundulus grandis, approximately 77 kilobases spanning AHR2A and AHR1A on chromosome 1, has swept to high frequency in resistant populations. WT is wild type, DEL is deletion. So here we've got a model of where AHR2A and AHR1A are in the genome. They're next to each other in the genome, separated by a gap here that's not shown. And there's a deletion that takes out the last exon of 2A and the first 6 exons of 1A in Fundulus grandis. It's around a 77 kilobase deletion. We see a similar deletion in Fundulus heteroclitus, the Atlanticode species, that takes out, it's in the same region of the genome, but it's got different breakpoints and it's slightly bigger. So it's a different deletion, but it's in the same region of the genome affecting the same genes. And in Fundulus grandis, this deletion genotype has swept to fairly high frequencies. So here is the wild type intact homozygote genotype. See that's at very high frequency in the sensitive populations. And here we've got the heterozygote and then the homozygote for the deletion. So we can see that in the resistant populations, the deletion is swept to very, very high frequency. Most individuals are homozygous. For the deletion, some are heterozygous, and very, very few have the intact AHR. We see this a little bit different in the intermediate populations, that the deletion haplotype is at intermediate to low frequency, and in the sensitive populations, it's almost not there at all. It's not there at all in S2 individuals that we sampled, and it was only present in the heterozygous form in a single individual in S1. Figure 3, evidence for adaptive integration from Fundulus heteroclitus. Panel A, sequence similarity dxy calculated between each resistant Fundulus grandis population and Fundulus heteroclitus, contrasted with similarity between a sensitive Fundulus grandis population and Fundulus heteroclitus. Negative values indicate similarity with Fundulus heteroclitus that is higher in resistant than sensitive Fundulus grandis populations, pink. Okay, so here we're taking the difference between the similarity of a resistant population and Fundulus heteroclitus compared to the similarity between a sensitive grandis population and Fundulus heteroclitus. The expectation is that either Fundulus grandis population should be equally divergent from fundulus heteroclitus. That's the neutral expectation. And that's what we see for most of the genome for each of these three resistant populations here. However, we do have these regions where the similarity between the resistant grandis and heteroclitus is much, much higher than the similarity between a sensitive grandis and heteroclitus. So if this number is much smaller than this number, then you get a negative value. And that's what we see here. It's deeply negative values at the beginning of chromosome 1 and another chunk on chromosome 10. Panel B. Genetic variation flanking the AHR deletion is more similar between resistant fundulus grandis and fundulus heteroclitus than between resistant and sensitive fundulus grandis, suggesting introgression. Again, this is just another way of visualizing kind of what we're calculating here. So here we're looking at genotypic similarity in multidimensional scaling space, and we're looking at the genetic variation that flanks the deletion. And what we see here is if you're from a resistant site, your black data, that you cluster with the genotype of individuals from fundulus heteroclitus. Whereas if you're sensitive genotype, then you're very different from heteroclitus or the resistant grandis. And the heterozygotes cluster here in the middle, as you'd expect. Okay, so this is consistent. This is unexpected, right? That the resistant genotypes should be more similar to a different species than they are to different populations of the same species. So this suggests introgression. Panel C. Mean time to coalescence of 1 megabase surrounding the AHR deletion between fundulus grandis and southern fundulus heteroclitus is 0.0026 mutations per site, and between intact fundulus grandis alleles and all of fundulus heteroclitus is 0.018. Interspecific coalescence times that predate population divergence, 0.0066, is evidence of gene flow. Okay, so basically this is what this is the amount of similarity you'd expect based on the divergence time, somewhere between here and here. But here we've got a much higher similarity for the deletion haplotype compared to heteroclitus than you'd expect if this was some function or feature of incomplete lineage sorting. Panel D. Large integration blocks are restricted to resistant populations. So here we're scanning the genome for large blocks greater than 400 kilobases that appear to be from fundulus heteroclitus, and we're looking for them in the genomes of individuals from fundulus grandis. So different block sizes, really, really big ones to sort of intermediate big ones down here, and we only find these heteroclitus blocks of DNA in the resistant populations. We don't find them at all in the sensitive populations. Panel E, maximum composite log likelihood ratios under integration and incomplete lineage sorting models as a function of strength of selection, pink bottom axis, and time between integration and the onset of selection, top axis, for the AHR deletion region relative to a neutral model. Integration is likely the source of the adaptive haplotype in fundulus grandis, which rapidly swept because of large selective advantage, S equals 0.8. Okay, so this is results of the model developed by Kristen and Sivan. You can see much more detail about this model that they developed for this analysis in the supplementary materials, including a lot of other models that they tested. But here we wanted to test what's the relative likelihood of this event, this integration event, being from integration or rather from incomplete lineage sorting. And as you can see, we've got much higher log likelihood of the integration scenario than the incomplete lineage sorting scenario. And the generations, the most likely number of generations to integration is very, very few. We're talking very recent integration. And the selective advantage offered by this integressed haplotype, and again, this is for the AHR deletion region, is pretty high. Selection coefficient that's most likely is around 0.8. So thank you for listening to this recording. Please have a look at the science website to find the supplemental material and all of the references and acknowledgements for all the people that worked on this project.